We have spoken about monosaccharides and disaccharides, and now we're going to talk about polysaccharides. Poly means many. So, and again, we've got the sac, that means sugar. So polysaccharides are made up of many monosaccharides joined together. Now, an important thing to remember is that polysaccharides are not sugars. So remember, we said monosaccharides and disaccharides were sugars because they taste sweet, they're soluble, and they form crystals. Polysaccharides are not sweet, they do not form crystals, and they are insoluble. So even though they are made up of monosaccharides, they are not sugars in themselves. We have 3 polysaccharides that we are focusing on. We are going to talk about starch, glycogen, and cellulose. And starch, glycogen, and cellulose are all made up of the monosaccharide glucose. But remember, we've got 2 forms of glucose. We have got alpha glucose, where the OH group points down on carbon 1, and we have got beta glucose, where the OH group points upwards on carbon 1. And this slight difference in the position of a hydroxyl group has this massive difference on the structure that the polysaccharides become. And if we divide it up like this, cellulose is made up of beta glucose, starch and glycogen are made up of alpha glucose molecules joined together. And if we just kind of summarize what we're going to be talking about, so starch is in plant cells, so plant cells only. It is for storage of energy, and it is made up of 2 different polymers. So it's made up of amylose and it's made up of amylopectin. Now both amylose and amylopectin are made up of alpha glucose, and we'll talk shortly about how they take on slightly different structures. Amylopectin is very similar to the structure of glycogen. So glycogen is again for energy storage, just like starch. However, it's found in animal cells. And it has a very similar structure to amylopectin, and again it is made up of alpha glucose molecules in that as well. Amylose will only have 4 glycosidic bonds. Amylopectin and glycogen will have 4 glycosidic bonds and 6 glycosidic bonds. And we'll come on to what that means shortly. Cellulose is made up of beta glucose molecules joined together. Again, like starch, it's in plant cells only. However, it is not for storage, it's for structure. And we'll talk about how it functions as a structural component of plant cell walls shortly. Starch, glycogen and cellulose are all insoluble under normal cellular conditions as polysaccharides. Glucose is soluble as a monosaccharide, and the reason it's soluble is all to do with the hydroxyl groups as part of the glucose molecule and their ability to form hydrogen bonds with water. So what we've got here, we've got a glucose molecule, and where you've got all these arrows like this, they are showing you where hydrogen bonds can form with water. And you've got the water molecules here and here, here and here. So this glucose molecule can form lots and lots of hydrogen bonds with water and is freely soluble in water. The polysaccharides starch, glycogen and cellulose are not soluble in water because they don't have these hydroxyl groups free to bind to water, to hydrogen bond to water. Instead, they will be hydrogen bonding either to themselves as a polymer or with other polymer strands, as is the case in cellulose. And we'll talk about that shortly. If we start with starch, like I said, plant cells only, mix of amylose and amylopectin, and is involved in energy storage. And what we've got on this picture here, this picture has been taken by just a light microscope, and it is a microscope image of potato cells. And what we've got, if I just show you an outline here, this is one cell here, and the purple structures that you can see inside, these are the starch granules. And if you think about potatoes, potatoes are filled with starch, and that's because they're storing the starch in these granules that you can see here. And these starch granules have been stained purple by using iodine, potassium iodide, to stain these cells here. So if we look at the structure of amylose and amylopectin in a little bit more detail, we have a greater understanding on the overall structure of starch and its function. So amylose and amylopectin are both alpha glucose. Amylose are long unbranching chains of alpha glucose molecules, and these alpha glucose molecules have all bound by the 1,4 glycosidic bonds that we were talking about earlier. So we've got a 1,4 bond here and a 1,4 bond here, and these are all 1,4 glycosidic bonds. So lots of 1,4 glycosidic bonds, and the bonds point in the same direction. So hopefully you can see they're all pointing downwards in this image. And this means it gives the molecule a curve. And when you build up hundreds and hundreds of these monosaccharides joining together, the molecule becomes helical. So it becomes helical like this. A helix is a very important structure in biology. We'll meet this structure lots, and amylose forms this helical structure. Amylose is insoluble in water under normal lab conditions because the helical structure hydrogen bonds with itself within the molecule like this. So it holds the helical structure together by all the free hydroxyl groups binding, hydrogen bonding to other hydroxyl groups within the long chain of the amylose, of the alpha molecules, alpha monosaccharide glucose molecules joined together, and it becomes helical. Amylopectin is a little bit different. So again, it's still only alpha glucose. So still absolutely alpha glucose. We still have the 1,4 glycosidic bonds, and they're still all pointing in the same direction. So we do still get this curve to the molecule, which will become a helix, just like with amylose. The difference is that we have branches coming off. So hopefully you can see here, this is a branch coming off. And what we have here is, again, another glycosidic bond, but it is not a glycosidic bond 1,4. It's between carbons 1 and carbon 6. So this is carbon 1, and this is carbon 6 of the other molecule. So you get these branches coming off. And we're going to get these branches every 24 to 30 alpha glucose molecules. So what will happen is you get this curve in structure and you get other branches coming off like this. If we look here, we can see another cartoon version of what this looks like. So again, remember they're both alpha glucose. Amylose is just the 1,4 glycosidic bonds, and you just become this helical structure. Amylopectin, you've got 1,4 bonds plus 1,6 branches every 24 to 30 alpha glucose molecules. that amylopectin has more ends. So if we look at the amylose molecule, we've got 2 ends to the molecule. If we look at amylopectin, we've got 1, 2, 3, 4, 5, 6, 7 ends to the molecule. And when we are forming this molecule and when we're breaking this molecule apart, remember we form this molecule by condensation reactions, releasing a water molecule, and we break this molecule down by hydrolysis reactions, needing a molecule of water. It means that amylopectin forms more quickly and breaks down more quickly. So we'll store glucose as amylopectin more quickly, releasing that glucose when it needs to. Both amylose and amylopectin are broken down. The hydrolysis is catalyzed by the enzyme amylase, and that will break it down to the disaccharide maltose, which will then be broken down by maltase into the alpha glucose molecules. And as I said, amylopectin is broken down much more quickly because it has more ends. Glycogen is again for energy storage, and it is energy storage in animal cells. So we will have it in our cells. And what you've got in this image over to the right is you have some liver cells from an animal. You've got the nucleus, where the DNA is kept, and all the pink material is glycogen. And so liver cells will store this glycogen, and it means you always have a carbohydrate source where when you need a release of glucose quickly, you can break this glycogen down by hydrolysis, release that glucose into your cells so that you can keep respiration going. The structure of glycogen is very similar to amylopectin, in that we have the 1,4 glycosidic bonds, and we also have the 1,6 glycosidic bonds. The difference is it branches more than amylopectin. So amylopectin was branching every 24 to 30 alpha glucose residues. Glycogen branches every 8 to 12 molecules. And what this means is, hopefully you can see it on this picture, this cartoon here. So we have starch on the left, mixture of amylose and amylopectin. Glycogen is just made of glycogen, more branches. Sorry. You've got more branches, and it means that you've got more ends even than amylopectin. So you've got all these ends of the molecule, and it's much more important that animal cells have quick release of energy than plant cells, because we need that glucose to power our heart beating, our brain, our neurons working, movement. So we need to have a much more rapid release of glucose than a plant cell. Both of these molecules, though, both starch and glycogen, the structure they become is very compact. And that's the key idea that we need for a storage carbohydrate, compact. It means that we can store a lot of energy in a small space. And that's really, really important. Another key idea is that they are both insoluble. Now this is important because if we had glucose in our cells, that glucose will dissolve in the cytoplasm of the cell, will dissolve in the solutions in the cell, and it will affect the osmosis, the water balance of the cell. And we'll talk about osmosis in more detail in the coming weeks. Both starch and glycogen are insoluble under normal cellular conditions, and this means that they don't upset osmosis. They're not going to upset the osmotic balance. They're not going to dissolve and wash away. They're going to stay where they're put. So, for example, if you're a potato and you're storing your energy in starch, that starch will stay in the potato ready to provide the energy to make new potato plants. Cellulose is different. So cellulose is made of beta glucose, and we can see a little image of beta-glucose to remind you. And remember, on carbon 1, the OH group is pointing upwards. Cellulose makes up the cell wall of a plant cell. So it's on the very outside of a plant cell. And we'll talk about this in more details in a few weeks' time. Here you've got a cartoon showing more details of a plant cell wall. You've got the cell membrane, and then outside of that you've got the cellulose layer that makes up the cell wall. And cellulose is made of long chains of beta-glucose molecules, all joined together with glycosidic bonds, those glycosidic covalent bonds that we've been talking about. We said that starch and glycogen become helical in their structure, and they are nice compact structures. Cellulose is different. Cellulose is going to be a straight structure, straight and strong. If you think about bridge building, engineering, in your building a structure that's strong, the structure will be based on straight lines. Straight lines are stronger than a helical structure. And the reason that cellulose becomes a straight molecule compared to starch and glycogen that become a helical molecule is all to do with the position of that OH group on carbon 1. So in this image on the left here, we have got two beta-glucose molecules next to each other. Now these beta-glucose molecules want to form a glycosidic bond between carbon 1 on one molecule and carbon 4 on the other molecule. And hopefully you can see that the OH groups don't line up, and that's going to be a problem with forming the molecule. So what happens in cellulose is that we flip one of the molecules over. So if we look over here, what I'm going to do is I'm going to highlight carbon 6. So this is carbon, sorry, I'll highlight it. This is carbon 6 on one glucose molecule pointing upwards. This is carbon 6 on the other molecule. So they're pointing in different directions. And if we highlight the oxygen group in the oxygen atom in the ring structure, you can see that they are pointing in different directions. And what this means is that we managed to line up the hydroxyl groups so that we can form that glycosidic bond between carbon 1 and carbon 4 very nicely. And this will happen all the way along the cellulose strand. Remember, each time we form this glycosidic bond, we'll release a water molecule, and we end up with the structure as seen on the bottom. And what we end up here is glycosidic bonds alternating in their position. So we've got a glycosidic bond down and up, down and up, down, all the way along the molecule. And this means that the structure is very, very straight. I often find it the easiest way is to kind of think where the carbon 6 is. So these are all the carbon 6 molecules pointing in different directions like that. So it means that this cellulose molecule will be straight and strong. What cellulose does to be such a strong and straight structure is it forms bundles with itself. What we've got on the image on the right here, this is a picture taken by an electron microscope. And it's showing you the, hopefully you can see the straight lines across the structure. You can see all these straight lines across it, forming this kind of mesh of cellulose. And this cellulose is strong, but it does let water through it. And that's going to be really important when we're talking about plant cells and how they behave under different osmotic conditions. The cellulose itself is not soluble, and it's not soluble, it's not going to wash away when it rains on a plant, because all those hydroxyl groups that would want to hydrogen bond with water, hydrogen bond within themselves. So what you end up with are different chains of cellulose. So here's 1 chain, 2, 3, 4 chains, all bundled together, and they all hydrogen bond with each other. So these chains bundle together, and they bundle together to form microfibrils, little fibers. They then bundle up to form macrofibrils, bigger bundles, and then they bundle up to form cellulose fibers. So very, 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 very strong. If you think about it as kind of a mind experiment, if you've got a pencil in front of you, one pencil, you could take that pencil and you could snap it and break it. It's strong, but you're stronger. However, if you take 20 pencils and try to snap them, you won't be able to snap them. They bundle together to be extra strong. And that's what cellulose does as a polysaccharide. So you've met three polysaccharides. Starch and glycogen store energy and are helical. Cellulose is straight and strong for structure. Thanks.